Go for it, Sophie.
Okay, so I have number 1, and it says basically to draw a DNA replication in the bacterium E. coli, and we have to have all of these things in it. And so we have the diagram right here, and so we have our topoisomerase or isomerase sort of down by the bottom. It's written on there, and then we have our DNA helicase, and it breaks the hydrogen bonds between the DNA strands. And then we have the single-stranded binding proteins, which are those little yellow balls, and it keeps the parental strands apart. And then we have the primase synthesis, an RNA primer, and that's the little red thing right here. And then we have the DNA polymerase 3 that synthesizes the daughter strand of DNA. That's a brown thing. And then the DNA polymerase 1 exercises the RNA primers and fills in the DNA. And then the DNA ligase covalently links the Okazaki fragments together. And that's it.
So yeah, if you're watching this, make sure you draw this out. Use your own handwriting.
A diagram here, but you can't really see it on the video, so.
Yeah. Go one more time.
What?
Go one more time.
Is it? There we go.
Yep. Like that. Draw it like that.
Yeah.
Or you can make it like a bubble. It can be
yeah.
like 2 bidirectional two replication forks. You can do that too.
All right, on to number 2.
All right, so I have number 2. So for number 2, it talks to the first one, A. This protein complex synthesizes primers in eukaryotes. It's DNA primase. And then for B, DNA A, this protein promotes the separation of the AT-rich sequences within the origin of replication. And then the epsilon subunit of DNA polymerase 3 contains the proofreading. Then DNA polymerase epsilon protein synthesizes the leading strand in eukaryotes. And it's the 2 sequence that dislodges the DNA polymerase 3 from the chromosome to terminate replication. And then DNA polymerase 1 is the protein that has 5' polymerase and 5' exonuclease activity. And topoisomerase 2 is the protein that introduces negative supercoils into the DNA head of the replication fork to stop the positive supercoils. And then the ORC protein binds directly to the ARS element, and the single-strand binding proteins prevent DNA strands from annealing during replication. And DNA ligase is an enzyme that seals Okazaki fragments into continuous strands for DNA.
Yep. Then we're on to number 3 with the
Okie dokie. So this one, the first one wants us to list at least 5 enzymes, prokaryotic or eukaryotic, that use ATP during DNA replication. So that's DNA ligase, MCM, DNA helicase, DNA A, DNA primase, and DNA gyrase, or topoisomerase 2. There's at least 5 enzymes, prokaryotic or eukaryotic, that consume deoxy ATP during DNA replication. DNA polymerases alpha, epsilon, delta 1, and 3. Epsilon. 3 or C lists 5 enzymes that have 3 to 5 prime exonuclease activity. DNA polymerase 1, DNA polymerase 2, and 3, delta, and epsilon. And indicate 1 enzyme, prokaryotic or eukaryotic, involved in replication that uses DNA as a template. That's synthesize RNA, that's DNA primase. And then this one uses RNA as a template to synthesize DNA, is telomerase.
Wait, on to number 4.
All right. So number 4 asks you to, you're trying to synthesize the DNA in the lab, and they give you the strand and the RNA primer. So the first thing you have to do is figure out where the primer matches up, and it ends up being that that last portion of the strand, which is reading it from 5' to 3' is CCTTTAAGCA. And then the next part with A, it's asking you to, well, it says you decided to perform DNA synthesis reactions, determine the sequence of DNA strands synthesized if you use the nucleotides described in parts A through D in your experiment, and assume that if the DNA polymerase cannot incorporate a nucleotide into the nucleic acid strand, the reaction will terminate after the last incorporated nucleotide. So the trick here is to know that to be able to do this, you have to have the triphosphates, which is the ones that end in TP. And so in number A, you have everything you need for the A, C, G, and T. So you end up with 5' ACT, GAT, GG, TCC, TGC, TAT, and 3'. And then B, you don't have the right triphosphate for C, and so you end up with 5' A and then 3' it ends there. And then C, you don't have the right stuff for the T, which UTP is an RNA, and so you end up with 5' AC and then 3'. And D is basically the same stuff, and you end up with 5' ACT and then 3'.
Yeah, which then brings it to me. I got number 5, and pretty much how number 5 goes, they give you a list of proteins involved in replication, and so they're listed here. And then it asks you questions and tells you to pretty much, of this list of proteins, figure out what goes where in the questions they ask. And so the first one is just asking which ones are found in prokaryotes. And so you have to go through and you have to look through. You have DNA A, DNA primase, DNA polymerase 1, DNA polymerase alpha subunit, DNA gyrase, and US are all part of the prokaryote, so around the bacteria. Then it asks which the bud proteins are found in the mouse musculus. It's a eukaryote. And so pretty much the first one was just what cells or what are in prokaryotes. This one's just what's in eukaryotes. And so you just go through MCM helicase, CDC6, DNA polymerase alpha, DNA polymerase epsilon, DNA primase, pCNA, and telomerase. Pretty simple vocabulary lesson. Study, write it down. The next couple are a little bit kind of tricky for me. I was able to get them through. Which of our proteins is involved in chromosome compaction? DNA gyrase, which is topoisomerase, if I'm correct. Correct me if I'm wrong, guys. And so this is just helps compact the proteins or the chromosomes. And then which of the above proteins involved in initiation stage of DNA replication? That's DNA A, CDC6. And finally, which of the above proteins involved in the termination stage of DNA replication? MCM helicase and TUS. So that is the group problem for this week. Thanks so much for watching, and we'll see you next week.